Hello everyone, today's topic is about the recent advances in bioengineering. We will cover CRISPR-Cas9 and then proceed to how to use it for purposes different from editing DNA, how many components they have, and how to best deliver them according to my experience in the field so far. Firstly, CRISPR, in the conventional sense, is a tool that can cut DNA and target specific DNA regions using a guide RNA. Therefore, it has become very easy to make for precise targeting multiple genes. However, the all-conventional CRISPR proteins are limited to a small number of precise edits and only in single cells. The reason is that each protein has a non-zero error rate, called its off-target rates, which add up to a significant value when used across large number of cells like in whole organisms. This is because organisms need well-tuned gene expression patterns across their tissues, where disruptions can lead to unexpected consequences. Therefore, its use in larger settings like in organisms such as human patients is not currently recommended. Since this precision is not 100% cutting in multiple places at once can cause too much cellular distress, especially in humans. The solution is to deactivate the cutting ability of the protein. This will enable targeting of specific genes of interest, but not hurt them in any way. Such a system comes in many forms for many uses. The most prominent and promising are the second-generation CRISPR-based gene modulators. These systems utilize 4 components which allow for more precise control over when the system is fully active. These components are the Cas9 protein, second its guide RNAs, third a guide RNA separator like CSY4 or tRNA depending on your guide RNA array size, and lastly a pair of gene activators and repressors that bind to the guide RNAs. The advantage is that each component can not only be further divided for more control, but also each one can be made to express only under specific conditions. For example, in cancer, there is typically 4 things I have noticed. The first is excess cell growth. The second is hypoxia signaling regardless of the presence of oxygen. Then there is inflammation, and finally oxidative stress and protein misfolding, signaling occurring simultaneously. These would typically be occurring separate or sequentially with respect to each other for most other cases. But, for added precision, a 5th input will be a good idea. In cancer detection, for example, the loss of active p53, an activated EMT suppressor SENAI1 activity indicates the kind of DNA damage sensing failure, and the repression of genes that stop epithelial mesenchymal transitions. Both are alarming signature of cancer. In short, healthy cells with functional p53 and without EMT are exempt from activating the circuit even if all 4 of the other inputs are on. To emphasize, when each input is engineered through the power of synthetic biology, they do so to turn on each component of our 4 CRISPR-Cas9 components. These I will collectively call a biocomputer due to its ability to sense what the cell is doing and then respond to conditions that are suspect by regulating gene expression. With this technology, we have a great opportunity to generate great medical advances to battle against cancers, since such systems can then target key drivers of cancer to stop them. Thus, finding the unique signatures of many systemic and local conditions is a great breakthrough that demands recognition. Much of the process is the same for other complex conditions like aging and defense against radiation damage while keeping tabs on any forming cancers. Some other uses extend to responses to extremes of temperature. Temperature extremes being a theme in climate change and one of the several major challenges facing humans, especially when it comes to overheating and freezing. Freezing being a concern for long-duration space travel, tissue and organ storage, and tactical responses to severe military conflict affecting Earth's surface. To address our overheating problems, one such solution with overheating is simply to stop the production of heat when outside temperatures are high enough to sustain the human and mammalian physiology. These challenges will define the 21st century to come. This is truly the defining moment in our history as new genetic discoveries are unlocking these new possibilities. The second breakthrough that is needed, however, is a way to deliver such CRISPR systems to people. Challenges persist in delivering these systems effectively. their size and required permanence in tissue, along with the limitations of conventional virus delivery mechanisms like AAVs and biopharmaceuticals targeting single genes, remain key obstacles. Additionally, the time and cost required to develop new therapies, coupled with immune responses to repeated dosing, further complicate treatment. AAVs, for instance, face immunological hurdles due to the immune responses they can trigger when reusing the few available serotypes, which get compounded from their lack of universal tropism and rare instances of genetic integration. To address these issues, I aim to develop a VLP-based biocomputer capable of detecting abnormal transcription factor expressions involved in cell stress. This system, when mature, will regulate gene expression in response to disease. However, foreign CRISPR proteins do still pose a significant immunological recognition risk as foreign agents, so I plan to compensate by developing a globally transducible VLP system, and there is yet another reason signifying this need. VLPs are virus-like particles which behave like viruses and are fully engineered piece of circular DNA capable of carrying and replicating our needed cargoes to all the cells of the body that need them. Due to global transduction, my VLPs may serve to reduce necrotic bystander cell death in stress conditions. This type of cell death, though shocking, is not really surprising. Basically, this phenomenon occurs when cells in a tissue are unevenly hardened against a set of stresses. This then causes some to die, leading to the release of enzymes that trigger a chain reaction of death to neighboring cells. This, of course, is solved by a global transducer system whose goal is to enter and protect all cells. Some key requirements, though, of VLPs of this category will be to be both functional and reliable and simple. Thus, I have designed two proteins to enable the replication of my VLP and its transmission. The first is a multifunctional packaging protein to enable transmission to all cell types through binding of molecules called integrins that are found on all cells in the body. Integrins are proteins that are attached to the membrane of all cells. They come in 22 combinations, as they are often bound to each other in pairs. Most are also able to bind small amino acid sequences on proteins that glue cells together through extracellular matrix proteins like collagens and laminins. Since all cells have them, and according to accumulated data, many of which integrin binding sequences are known, it is possible to us to design our needed receptor protein for the needed VLP. Next, the VLPs will need a heavily modified viral protein that can bind cellular DNA but not integrate with them. Such is the case for a protein from the common Epstein-Barr virus, EBV protein, which enables genome surveillance and regulation called EBNA1. This protein is responsible for replicating the episomes of EBV and anchoring them to chromosomes while also suppressing key antiviral and cell cycle suppressive proteins. Therefore, these antiviral processes are mutated away. In addition, its ability to regulate gene expression is silenced amongst other changes. But why should future VLPs target integrins for universal transduction? The answer is to allow the needed mobility for VLP to move to most tissues as stated earlier. In doing so, they are able to enter the body's immune cell training centers located in the thymus and bone marrow. This then will allow the immune system to accept foreign proteins as self-molecules through the training of new VLP-insensitive T and B cells in the thymus and the bone marrow respectively. This is also called immune training by positive and negative selection of cellular peptides for establishing self-recognition. However, one caveat is that time is needed to create new immune cells, and in that time it may still require patients to be on temporary immune suppression. This will also hit older people harder, as they have harder time remaking their T cells due to the disappearance of their thymus. This therefore promotes the need of fixing the loss of the thymus with age, which will inevitably boost longevity. Global transduction offers enormous potential for safe delivery of gene regulatory pathways and gene circuits, to senolytics and gene replacement therapies, and much more. These technologies offer solutions to global challenges such as climate change, demographic shifts, and healthcare burdens. Therefore, VLP cargoes could be tailored to specific therapeutic needs in practice. Finally, all VLP design must include an ejection system. This not only satisfies the personal choices of end users, but also acts as a safety net and removes the concern of permanent stranding by its design should a VLP become damaged or in need of replacement. Of course, current VLPs are limited to one ejection system. Thereby the need for the discovery of more systems will grow, which will enable VLP selection over blanket ejection. Since all VLP systems must contain the ejection system, we will now look at the currently available option for this key feature. Our system utilizes a common and popular bacterial and yeast-derived antibiotic switch called TET-ON and a yeast-unique transcription factor system called GAL4-UAS, added to it for positive self-enforcing feedback. Self-enforcing feedback is needed to enable a single dose of tetracycline to activate our removal system. The single-dose method can then better ensure that tetracycline isn't abused by patients, which would otherwise lead to potential antibiotic resistance epidemics. However, this will mean that patients are unable to utilize this antibiotic or its derivatives for medical use and must declare this to their doctor if they wish to keep their VLPs. For everyone's reference, TT is tetracycline, which is an antibiotic that is safe for human use, while GAL4 serves to lock in positive feedback when any removal command is sent. Therefore, at present, tetracycline is perfect for administration for VLP safety systems for inhibition of its replication and receptor production by activating antisense-encoded truncated egress proteins. This is intended to ensure that upon tetracycline administration, the secreted VLPs cannot bind to cells when instructed by the consumer to leave, thus enhancing the system's safety, exchangeability, and flexibility compared to existing technologies. In closing, the recent scientific advancements in cellular stress responses have accelerated therapeutic research, particularly in cancer treatment. The human understanding of how cells react to stress has vast potential to improve cancer survival, longevity, and reduce healthcare costs. Recent biotechnology innovations, including CRISPR-based biocomputers, offer new modalities for health, but they still remain on shelves due to the delivery issue, which I would like people to consider supporting the development of. This kind of research aims to introduce DNA-binding RNP biocomputers into genomic medicine and biomedical engineering. Initial proof-of-concept studies will be hopefully quickly followed by further research involving gene targets in animal models before human trials. However, the number of regulatory steps is greatly dependent on the immediate need of the product being developed. The ultimate goal for humans now is to develop a global transcription factor-based sensing and response system to address complex issues such as cancer and age-related diseases, including novel enzymatic pathways and gene regulation. Then, in consequence, by also overcoming immunogenicity concerns with globally transducing systems, we can expand the scope of biomedical discoveries and applications to improve living standards in an age of dynamic changes. And that concludes this video. Click like and subscribe if you want to know more on this new promising technology and its development. Until then, see you next time.